Good afternoon, everybody. What would we do without ITs? Thank you, Nunve. Before presenting your next speaker, please allow me to tell you a very short story to put things in perspective. Way back, working in Africa as a veterinarian, I was very concerned with parasites because they were particularly nasty for domestic animals, not so many for wild animals. But the only parasite that really concerned me personally was malaria, because it would floor me at least once a year. But I was not very scared of them. And then I got to know José Costa, and things changed dramatically. I think he'll have time to show you a little bit, but they are much more complex than I thought, from parasites that produce chemicals that activate estradiol receptors. I thought that was crazy. To parasites that can do more damage to your liver than one bottle of whiskey per week. So I got to know with him, to work with him. He's senior researcher in the National Institute of Health, always worked with parasites, did several discoveries during my time working with him, fantastic for the country and internationally. He is also an excellent manager of multidisciplinary research teams. He's director of our Center for the Studies of Animal Science, has been very successful, and does, without further delay, the floor. It's all yours. Thank you.
Thank you, Antonio. Okay. Thank you, Begonia. Begonia, you were very helpful in all these days. Thank you also to the One Health group at ICVAS. And thank you for this invitation. I thought? Oh my God. So, let me tell you that One Health initiative is something very useful for me, particularly as scientific coordinator of CECA, because when I was obliged to make a restoration to reform our unit, the concepts derived from One Health were very useful for us. This is congratulations for this school to choose this subject for a meeting like this. I will choose, we discussed in my unit, what kind of talk do you present in the One Health Day at ICVAS? And many of them choose me, and they said, you go to speak about liver fluke parasites, because antibiotic resistance, because microbiology, there are a lot of people talking about this. So nobody talks about this kind of parasites, this kind of beasts that are really very dangerous for a lot of people all over the world. Liver fluke parasites. I choose this title from my fellows in Thailand, Without Borders, Parasites Without Borders, from the bench to community. This is good, this is nice. Oh, pictures that we will see, it's a kind of movie that we'll see later. What are you talking about this day? This text from the authors of the One Health concept. Oh my God, everybody is agree that we need to accelerate, we need to work together in collaboration for accelerating biomedical research discoveries, enhancing public health efficacy, expanding the scientific knowledge base, etc., etc., etc. And the authors repeat, when properly implemented, it will help to protect millions of lives all over the world. Thinking about this, I thought to me, as scientists, can we act alone? No. When properly implemented, and the problem is by you, who are the responsible to implement the things that we found in the laboratories? Let us say if we arrive to a common conclusion. Well, I talked to you about this. Oh my God, excuse me, these technologies. These three beasts. First one, amphimaries. All them are zoonotic parasites. All are hermaphrodites. That means male and female inside the same body. They have a complex life cycle, but with many similarities. Amphimaris, Clonorchis sinensis, and Opistorchis vivarini. Very similar, but completely different in the outcome of clinical situation when infect human patients. Amphimaris was thought to be a parasite for animals, dogs, cats, etc. In the beginning of the second decade of this millennium, in Ecuador, you can imagine, in the middle of Amazon, some friends of mine described the first human cases among Indian tribes. Most probably, according to them, Manuel Calvupina and Williams, probably is not carcinogenic. But probably these are carcinogenic, recognized by IARC as definitive causes of cancer. And what kind of cancer it promotes? Cholangiocarcinoma, a dramatic cancer, a rare form of cancer in Portugal, but a very complicated situation all over the world. Carcinogens. Why some parasites are carcinogenic and others are not? This is the question that we tried to respond working a long time ago. We read, many of us, Thai people, Portuguese and Americans, we write many things about this. But the question is, this inconsistency, that means why amphimaris or Fasciolepatic are not carcinogenic and why Opistorchis vivarini or Felineus or Clonorchis are carcinogenic. This is our main problem. This is the life cycle of Opistorchis vivarini. It's here only to tell you that humans or animals became infected by eating this metacercaria. The metacercaria is inside of skin or the flesh of the cyprinoid fish. This metacercaria, you can kill it. That means you can interrupt the transmission if you put the metacercaria at 60 degrees Celsius during 5 minutes. That means a little time in kitchen and the fish were very good to eat. The problem is that Thai people and in East Asia, they eat this fish raw fish. Let me tell. Okay, clinically is usually Clonorchis or Opistorchiasis, always the same, and cholangiocarcinoma in the chronic phases of infection after 7-8 years of recurrent infections. This slide is from a Thai friend, and he loves to put nitrosamines as responsible also for inducing carcinogenics. I do not agree, but the authorship of this slide belongs to Winchell. These two parasites, Clonorchis sinensis and Opistorchis vivarini, or three, and Opistorchis felineus, they are very spread in all Russia. The Russian Federation, Opistorchis felineus, China, Correia, Indochina, I do not know if we can talk about Indochina, but there are millions of people at risk to get infected by eating raw fish. Raw fish from the rivers, not from the sea. Look to this poor fisherman in Thailand, in the city of Khon Kaen district. He gets the amount of proteins to feed his family. And they make this kind of plate, koi pla. Everybody eats this. What is this? It's the macerated fish at home and mixed with vegetables and spices. It's really, they are very nice. When I to eat quite a lot, I said, no, thank you. Thank you very much. But everybody eats this. They eat koi pla and they get infected. This is the hospital of Khon Kaen, where the story arrives in this form. This is a lady infected with Opisthorchis and cholangiocarcinoma. No treatment. We do not know any kind of treatment except the bistoury of a surgeon. Look to the registry, tumor registry, liver cancer and all other forms of cancer. Liver cancer is really, they have more or less 10 million people infected and with cancer, with cholangiocarcinoma in Thailand. It's all human Portuguese population. So one day in Thailand, in Khon Kaen, I decided with my friends in Russia, the United States, and Thailand, to make an experience. Using this, we cannot elevate or produce metacercaria at laboratory. So how you get the metacercaria? By fishing, catching the fish in the rivers, and collecting one by one the metacercaria. Fish in the rivers of Siberia can have 1,000 metacercaria. Maria and Slava, they get 50 metacercaria to infect hamster during 13 weeks. The experiment during 13 weeks. That means in Novosibirsk, in Siberia, they infect the hamsters, and after 13 weeks, Maria and Slava, they kill the hamsters and they bring to Porto. Okay. And they bring samples of urine, blood, and liver to Porto. And the things that we're going to see next, we're doing here at my laboratory, with some machines of the University of Porto, and at the hospital, the Pedro Spano. Look at this. This is staining hematoxylin eosin of a liver, and the staining, the Manson trichrome staining to see the fibrosis. No evidences of inflammation or fibrosis in these control hamsters. Teresina Amaro, at the hospital of Pedro Spano, they use this kind of classification table to define the biliary intraepithelial neoplasia according to this kind of histological feature, bilin 1, bilin 2, bilin 3. Bilin 3 is almost some, we can talk about a precancerous niche precancerous. It's not a tumor, but it's over, it's running to this situation. Look at this. The parasite is here inside. Look to this black holes. Is emoswine, like the Plasmodium has emoswine. Opisthorchis viverrini, for instance, do not have emoswine or emoswine. You see a huge inflammation, a portal area enlargement in hematoxylin eosin, staining with Manson trichrome, this fibrosis, a huge periportal fibrosis. This is really very remarkable because but it's necessary to look more near to the situation. Look, we can see mitosis, signal of mitosis, like this here. You can see a building 2, a building 3. This is the inflammation, fibrosis, the cell disarrangement is so big that we classify, that means the histological people classify this like a pre-cancerous niche. This is a immunohistochemistry P63 to show the cell proliferation of the adult cells, build-up cells. It's huge proliferation, a situation of building 3 and some agranulomas. Look, this is a group of histologists who classified this. This is a revision. The Japanese, Europeans, Americans, they get this kind of table to define this situation that for us is really very important. Building 3 is an important step in order to cholangiocarcinogenesis. The worms, when we collect the worms inside the livers of the hamsters, we destroy them and we put them in the machine, LCMS analysis. And what we have here, these are bile acids in different forms, chemical forms of bile acids. Look, you have here the parasite, look to the eggs, and the eggs inside of liver parenchyma, here and in other points. We collect the eggs by destruction the liver, and look to this. What you have here, oxysterols, that means metabolites of cholesterol, but here, the bile acid is as a new feature. What is this? Nitrogen bases from DNA, usually adenines or guanines. That means that the adducts, we call them, the chemistries call them adducts. That means that a reaction during the staying of eggs inside of the liver, they exhibit what is a signal of oxidative DNA. That means these bases, these nitrogen bases, they were captured from the DNA chain. This works similar with our model in schistosoma hematoma. And Mr. Cavalier and Zaid from the Nebraska Center, Cancer Center, medical in Nebraska, in the University of Nebraska, they proposed a situation like this. The schistosoma produces a lot of metabolites of estrogen. And this estrogens by coenzyme P450 is activated to the form of catechol. These catechol forms, these metabolites, they have the ability to react with DNA and to take an adenine or a guanine, nitrogen residue. What happened to the DNA? That means there's an aporinic site. This is a possibility to the beginning of mutations. What are the most frequent mutations that you have in certain genes? The gene TP53, that means the gene who expresses the P53. And in the patients associated with herpes virinae, and also in people infected with schistosoma hematoma. So, estrogen metabolites or oxysterols metabolites produced by the parasites, and they have in common the ability to react with DNA, oxidizing the DNA and promoting eventual mutations. So, in a publication, we make the reunion of two things, schistosoma and Opisthochus vivarini, and Phelinel's is similar. Here you have the human chromosomes. These oxysterols, or bile acids, and the metabolites of estrogen, they have the ability to react with DNA. If there are any problem with the reparation to repair DNA, you can have mutations. Associated with inflammation and fibrosis, you arrive to a point of precancerous niche and possible in the future with two more steps, a carcinoma. This is a picture from Thailand, from Correia. You see here, you have the Opisthochus vivarini, and this is a cholangiocarcinoma for a patient. This is a spinocellular carcinoma in the bladder. Why certain parasites can are carcinogenic and others do not? Because this. When you put Opisthochus vivarini, Opisthochus phelinells, and Fasciole hepatica together in the machine, in the same, using the same systems, look to Opisthochus, the oxysterols are here. Much greater things in Opisthochus vivarini. Phelinells, you have the oxysterols, but in less quantity. Look to Fasciole hepatica. No bile acids at all. This is the worm, the entire worms. I have here a space for amphimeros, because I think as soon as I receive the amphimeros from Ecuador, because I think and I believe that amphimeros, it's only infecting humans for 10 years. It begins in 2011, so we have 11, 12 years. It's not much time to get cancer. So I think if amphimeros infection in Ecuador and in other countries in South America, if it is not linked to carcinoma, to cancer, no problem. Cholangiocarcinoma or other form of cancer. So I believe that amphimeros is similar to this situation of Fasciole hepatica. You can read, if you're interested, those publications where we talk about the experiment with Russians, anti and Americans, etc., and a system that, even if this hypothesis that we have described, if you are right, can you help the patients with this? No, you cannot do nothing. Oxysterols, what are oxysterols? You can do nothing. But this is for synthesis. I'm sorry, but I like a lot of Beatles, and Beatles, sometimes some songs illuminate us from the dark black night, working at a laboratory. We don't know nothing about nothing. And sometimes it's necessary to listen, good music, to go on. And this is a picture of Dr. William. He gets his PhD thesis in Salamanca, and I was happy to stay there discussing this thesis. William promised me to send me a worm of amphimeros, but it didn't come yet, with quarantine and things like that. Amphimeros was thought to infect only cats and dogs, now infects humans. Look to this picture. Is a woman from Ecuador from this part of the country, nearby Amazon, and look to the news today about the wars, drug wars and cartels, etc., and the political instability and poverty. And this intimate relationship between humans and animals is critical also. It's necessary to fight against poverty, but all over the world. Stop. Another thing, probably some of you have listened about Fluke Parasites. Is a trematode, hermaphrodite, no, not carcinogenic, and it's probably the most important parasite in Europe, but nobody cares about it. This is the liver of a cow, a cattle, I think, the ducks, biliary ducts, and the worms there. Usually we go to the slaughterhouses to get this kind of material. We get also eggs, and in the laboratory, we make interesting things like activate the existence of miracidium. The miracidium infected the snail, a limnaea. Inside of limnaea, they divide in radius, first generation, second generation, until Cercaria, like this, and finally a metacercaria. We do this in our laboratory. Unfortunately, we cannot do this to produce Opistorchis folineus metacercaria or Opistorchis viverrini metacercaria. Life is like this. Is hepatic importance in Portugal? And how can we help people, the farmers? We choose a region in Vagos, in the south of Porto, because it's not the incidence or prevalence of the fasciolosis is not expected to be enormous, huge. And this work has 20 years. We have done this 20 years ago, and nothing happened. You know the landscape, look to the natural organic fertilizer, that means shit of the cows, to put in the fields. You know, many of you know about this. And look, we try to start during almost 3 subsequent years the seroprevalence of fasciolosis in Vagos. And look at this, in the first year, first semester of 97, 11%. That means in each 100 cows, 11 were infected. But in middle of the second part of the year, in 98, rainfall was really very, very important. And the incidence or prevalence is higher, much more higher, until almost 50% of all cats were infected. Can we use and help the farmers to have better results controlling the fasciolosis? Yes. Look at this. During the 3 years, a PhD student of mine in my laboratory, Antonio, we're now working in Coimbra. He collects snails in the environment. He brings the snails to the laboratory. We dissected the snails to see if they were infected. And infected snail, non-infected snail, and we get this kind of using the temperature and the rain, water and the temperature in Vagos region, and using predictive models, we get these 2 peaks. 97, you have shorter peaks. What this means? This means that the infected limnaea in environment is excreting, is emitting Cercaria. That means the infection begins to happen. When cattle goes to the fields to eat his food, it became infected because the Cercaria begins in April. or when it rains a lot in moss. Always two peaks. So what is the best time for the farmers to give ivermectin or other parasite to treat the cattle? Is not any part in the 12 weeks. The correct moment is this one, April, March, and April. And the second one in August, September. Not every year. They can, the costs of the production, they will be better. And the efficiency of the drugs became much more important because ivermectin is able to kill the juvenile forms, is not able to kill an adult form. Another situation. We have a lot of recombinant antigens expressed in my laboratory. And one of those, F8, like this is the structure, is an allergen. We can use this to make intradermal reaction. Look what up. Experiment in Coimbra was not like this. An injection with 10, 20, 14 milligrams of protein equivalent of this recombinant antigen. And look what happened 1 hour and 1 hour and a half later. This is a negative control, this is a positive control. That means a farmer can do this in the farm. During a morning or a day, he can know everything. It's not necessary lysis, Western blot, immunofluorescence, or another thing. An injection of now, this is more pressure now than 20 years ago. This is why we cannot use intradermal reactions to treat, to separate infected cows, non-infected cows in a farm, and the infected cows are treated, the non-infected cows are not treated. We don't need treatment to them. We can do this. So have you a model in vivo for cholangiocarcinoma? I think so. The Russians also, and Americans who work with us also. Opisthochus filineus is yes, Opisthochus filineus, we propose, is a parasite grade 1 carcinogen, not grade 3. Oxysterols are possible biomarkers of cholangiocarcinoma, probably, but we need to work a lot in this, and this is not easy to work. Intradermal reaction, it's necessary. Yes, it's not in fashion, but is very, very important. We can use in a better way the parasiticide drugs. And let me tell you that combined therapy, if Planorbis sinensis or Opisthochus viverrini are carcinogenic, we cannot treat them with the same drug used for other parasites, like praziquantel. We need much more things because these people, they're going to, with recurrent infections, they will be a cancer patients later. So we need to have a combined therapy. And what I'm working now at this moment, using antioxidants and immunotherapy. What for? This here, to block oxidation of the DNA of the hosts, to impeach the formation of adducts. That means to impeach, to down, indeed, in reality, to impeach the oxidation of DNA. Can we do this? We are working now. This is my group at CECA and INSA, and this is my fellows all over the world, a lot of them in Russia, because this is a picture from Russia, and a lot of people working with us, and thank you.
Thank you, Joseph. I think he may have convinced you that not only viruses and bacteria can be pretty damaging. So the session is open for questions, please. One here.
Thank you so much for this so interesting presentation. I learned a lot. I have two questions, if possible. The first one is, I wonder if it's possible to act preventively before the infection reach the animals. I mean, somewhere in the environment or between the environment and the animal. This is the first question. The second question is, we are now fearing more of this sort of...
Without the mask. Because I did that the studio at 50%.
Sorry, it's better now?
Oh, yeah.
So the first question was if there are ways of acting, for example, with Fasciola hepatica, if we can act in the environment to prevent the animals to be infected, or somewhere between the environment and the animal. This is the first one. Sorry, I'm a little bit bad of the throat. The second is with climate changes, probably we'll have a big rise of these problems. Probably they are already going on. We don't know. Here in Portugal and in all South Europe. So what do you think about it?
Second part, I don't know nothing about the climate. I talk too much about that. Everybody is
You
a
believe on
specialist.
it.
Excuse me?
But you believe on it.
Yeah,
Okay.
yeah, I do. I believe, but I do not know nothing about that. Hypothesis 1 degree, 2 degrees, I do not know. I only know what I see. That means what I try to understand. Okay, so I do not know what to tell you about 20-something years ago, a professor here invited me to think about climate change, what happened to schistosoma. That means to infections transmitted by vectors, by smell, like Fasciola hepatica. Fortunately, the restaurant is about that. So, no problem, no problem. I don't remember what I wrote a lot of things, and many book was published, a book. I only know that every quarrel talk about our we need to have monetization system, different systems from what I do until now. Times are changing. We have new problems, and we can have the same solutions. We need to think about this. In this point of view, let me tell you that we need to. We have to think about things with other people. They know much more than I about anything. So what we need to do is to put together people who know about the climate change and think together to do? What can we do to help them? What can we do to fight for a better world? What can I tell? Climate change is in today. It's a big problem. I don't know what to do. I hope that the temperatures do not. I do not work with interactive computers. I do not work with above 28 degrees. I'm nothing. I'm like butter. What can I tell? The first question is what to.
I ask you to be very brief. I think if I can put the question is if there is a way to prevent it before the animals get infected.
No. The only thing we can. Well, 1 year, many in the last century, in my laboratory, those times we do diagnosis of tumors. And one day, a technician in the office and tell to me, José, we have 12 people from the same city, from the same village, infected with Fluke Parasites. Oh my God, 12 is a problem. We call the health center, and we decide to talk with them. They didn't know because the patients were treated in Guimarães, in Porto, etc. So the local public services, health services, they have no conscience about this information. So first, to talk with the local people and introduce the problem. With we talk about with GNR or with a lot of people. We during 1 year we make a situation evaluation to stop encapsulated the body. A physician from the center, the service center, he talked to the television. Oh, Fluke Parasites is a beast, can kill people. Oh my God. What happened the next day? All the farmers in Rome, loads, they stopped to sell watercresses. And they came to the television to say, we need help. We need, because in the north of Portugal, they discovered a beast, and the beast stopped our economy. And 2 days later, I was called to Vila Franca de Xira to help the people, the watercress. We went there and we collect snails. That means a lot of us from my laboratory in the land collecting snails, Linnaea and all kind of snails that we found in the land. The farmers, they look at us. What are they doing? They are collecting snails. Oh, snail? What for? It seems that snails are the responsible for these parasites. Okay, no problem. During the night, you know what they have done? Tons of obviously calvin, oxytocin. And they put is a molluscus, and they kill all kind of mollusks in the land. Okay? No. But people do it. People do it. Many things. I do not know exactly what to do. I try to. Those people who work with me, we try to have good tools for diagnosis, good tools to diagnosis like in the cattle, in the human, etc. But I do not know how to prevent. Prevention is do not eat watercress, wild watercress. If you use wild watercress, you boil them in soup, not in salads, and it's necessary to be portioned. That's right.
Joseph, let me get one more question. We are already out of our time, and I'm sorry, this will be the last one.
Thank you, Zé
He'll be around.
You have to answer quicker the response. Well, thank you, very interesting. I remember a clinical researcher I worked in '92 in Israel. He used to say, he's a immunologist, he used to say everything in medicine is autoimmune until proven contrary. But last years, he says everything is infectious until proven contrary. And you demonstrated it clear. But so the question is, the immune system is not doing its job, preventing the infection and preventing the carcinoma development. And so I know that these parasites have a lot of escape mechanism, but my question is, there is a role for inflammation. And I think everything is inflammation until proven the contrary. Yeah, so there are a lot of inflammation here. And you put there immunotherapy. Who do you suggest to treat with immunosuppressors? Those? No?
Carlos, we are just in the beginning of this tour. Later, well, we can go there and I tell you about this. Indeed, what we're trying to do is very simple. The pharmaceutical oxysterols like cysteine or everasol can bacteria and put in cells and we published. I can give you the publication and you read. Second, these people usually, that's Bunshop, this guy here, Bunshop, now he's retired. Bunshop trying to explain to those Thai people, do not eat raw food. Because it's in the culture, it's tradition. They talk together, it's impossible. I tell you. You go to Mozambique, they have a lot of sardines. They have sardines, not good. And you give the sardines free. No, without any payment, because they are very poor. That tissue must be very, very, very poor. So, what can you do? Usually they are malnutrition. So, can you help? Can you help them? Can you stimulate the immune system? Yes, I see so. And we are working with some antigen there. We are driving experiments. That model, hamsters infected with opisthosis. But we need to work a lot of time. This is just, we are just in the beginning. But I have some good indicators that maybe you are correct.
Thank you, José Manuel. I think you have scared a few more people about parasites and diseases in humans. Great presentation. Thank you very much.